Part 2, the Omega System. Chapter 4, what Omega-3 and Omega-6 really are. Omega-3 and Omega-6 are usually presented as if they are supplements, categories on a nutrition label, or opposing health teams competing for moral approval. That framing is too crude to explain what they actually are. Omega-3 and Omega-6 are families of polyunsaturated fatty acids defined by the position of the first double bond relative to the methyl end of the fatty acid chain. That chemical distinction sounds small, but it changes how the molecule behaves inside the body, how enzymes process it, what kinds of membranes it helps build, and which signaling mediators can later be derived from it. These are not just fats in the dietary sense. They are structural and signaling substrates that enter phospholipids, alter membrane behavior, and later serve as raw material for inflammatory and pro-resolving chemistry. To understand them correctly, they have to be pulled out of the supplement aisle and put back into physiology. The reason these fatty acids matter is not simply that they are polyunsaturated. It is that the body uses them as part of its surface architecture and signaling reservoir. Once incorporated into phospholipids, omega-3 and omega-6 fatty acids become part of membranes where they influence packing, flexibility, oxidative susceptibility, enzyme access, receptor environment, and the kinds of lipid mediators that can be generated when the cell is activated. This means they are not passive inputs. They shape the interpretive surface of the cell and the chemical options available once signaling begins. A membrane containing different proportions of omega-3 and omega-6 is not just chemically different on paper. It behaves differently under stress, produces different mediator profiles when phospholipids release fatty acids from a membrane, and changes how easily inflammation escalates and how effectively it can later resolve. Omega-6 fatty acids are often discussed in a distorted way because they are associated with inflammatory signaling, but that association gets oversimplified into the idea that omega-6 is inherently harmful. That is not biologically accurate. Omega-6 fatty acids are normal structural and signaling components of human tissues. Linoleic acid is the main dietary omega-6 fatty acid, and from it the body can produce longer-chain derivatives such as arachidonic acid, which becomes one of the major membrane substrates for eicosanoid signaling. Arachidonic acid is not a mistake. It is part of how the body mounts acute responses to injury, infection, vascular disruption, and local stress. Prostaglandins, leukotrienes, and thromboxanes derived from arachidonic acid help control blood flow, pain signaling, immune recruitment, platelet behavior, and multiple forms of defensive activation. The problem is not that omega-6 exists. The problem begins when its structural and signaling dominance becomes disproportionate, when initiation chemistry outruns resolution, and when the body builds membranes that are more prepared to escalate than to finish. Omega-3 fatty acids are distorted in the opposite direction. They are usually marketed as anti-inflammatory nutrients, as though their main role is to suppress symptoms. That is too shallow. Alpha-linolenic acid is the plant-derived short-chain omega-3, while EPA and DHA are the longer-chain omega-3 fatty acids most directly involved in membrane function and specialized signaling in human tissues. EPA and DHA matter not because they are universally calming molecules, but because they alter membrane composition and provide substrate for mediator families that help shift inflammation toward completion. EPA can contribute to eicosanoid competition and to the generation of E-series resolvins. DHA contributes to membrane properties, especially in neural tissue, and serves as a precursor for D-series resolvins, protectins, and marazins. These are not simple anti-inflammatory agents in the blunt sense of shutting the immune system down. They are part of the machinery that helps the body stop recruiting unnecessary damage once defense has served its purpose. Omega-3 therefore matters less as a suppressor than as a participant in better inflammatory sequencing. This is why omega-3 and omega-6 should not be understood as opposites in a simplistic nutritional battle. They are parallel substrate families entering overlapping structural and enzymatic systems. Both are polyunsaturated. Both can be built into membranes. Both can be mobilized from phospholipids during signaling. Both influence the physical state of the membrane. Both compete, directly or indirectly, for elongation, desaturation, incorporation, and downstream enzymatic conversion. The real issue is proportion, placement, turnover, and context. When omega-6 derived substrate dominates tissue composition and inflammatory tone is chronically high, the body tends to generate a signaling environment more heavily biased toward initiation and amplification. When omega-3 incorporation improves, especially in the presence of better overall lipid stability and oxidative control, the membrane environment and downstream mediator pool can begin shifting toward a more balanced inflammatory pattern. But even that statement needs care. Omega-3 does not erase the need for omega-6, and omega-6 does not automatically create disease. The deeper question is what kind of membrane and what kind of signaling reservoir the organism has built from the total lipid environment it has been given. The chemistry also matters because these fatty acids are more fragile than more saturated lipid structures. Multiple double bonds create flexibility, but they also create susceptibility to oxidation. This makes omega-3 and omega-6 biologically powerful and biologically vulnerable at the same time. A membrane enriched in polyunsaturated fatty acids can support dynamic signaling behavior and provide a broad mediator substrate pool, but if oxidative protection is weak, if inflammatory load is high, or if membrane repair cannot keep pace, those same lipids can become sites of peroxidative damage. This is one reason the conversation around these fats becomes misleading when it is reduced to simple intake targets. The value of a polyunsaturated fatty acid depends on the membrane it enters, the tissue using it, the oxidative environment surrounding it, and the broader lipid architecture that stabilizes or destabilizes its presence. Flexibility without protection becomes fragility. Tissue specificity adds another layer that is usually ignored in mainstream discussions. DHA is especially important in neural tissue, retinal tissue, and other highly specialized membranes where curvature behavior, receptor function, excitability, and long-horizon structural maintenance depend on very particular lipid environments. EPA is often discussed more heavily in relation to circulating inflammatory balance and mediator generation. Arachidonic acid remains central in immune and vascular signaling because rapid local inflammatory response still requires substrate for initiation. Different tissues therefore use omega-3 and omega-6 differently, and the body is not trying to make every membrane composition identical. It is solving tissue-specific structural problems. The brain does not need the same membrane behavior as a neutrophil. The retina does not need the same signaling timing as a platelet. The intestine does not need the same lipid surface as skeletal muscle. What matters is not a universal ratio imposed onto every cell. What matters is whether the organism is remodeling tissues toward a coherent and functional lipid distribution rather than toward chronic, interpretive instability. This is also where the distinction between dietary precursor and tissue-active form becomes important. Short-chain plant omega-3 in the form of alpha-linolenic acid is not the same thing as preformed EPA and DHA in terms of tissue effect because conversion is limited and variable. The body can elongate and desaturate alpha-linolenic acid, but in many people the production of EPA is modest and DHA is even more constrained. Linoleic acid conversion toward arachidonic acid is also regulated rather than automatic, but the modern environment often supplies omega-6 substrates so abundantly that tissue competition shifts over time. This means the question is not merely whether someone consumes omega-3 or omega-6 in any form. The question is which forms are entering tissue, how much competition exists at the enzyme and membrane level, and what kind of long-term remodeling that intake pattern creates. Nutritional labels flatten this into grams. Biology turns it into structure and signal probability. The phrase essential fatty acid comes from this reality. Certain polyunsaturated fatty acids or their precursor classes are called essential because the body cannot synthesize them de novo and must obtain them from diet. But essential does not mean unlimited and does not mean that all downstream outcomes are automatically beneficial. It means the body needs access to these lipid families because they are part of membrane construction and signaling capability. Without them, the organism loses important structural and communicative functions. With them in excess or imbalance, especially in unstable metabolic conditions, the organism can build membranes that are chemically reactive, signaling heavy, and difficult to regulate under chronic stress. The same feature that makes these fats necessary also makes them capable of distorting physiology when proportion and context are lost. That is why they have to be understood mechanistically, not ideologically. What omega-3 and omega-6 really are, then, is not a pair of health trends or a ratio to memorize. They are families of membrane-active, signal-generating polyunsaturated fatty acids that shape how the body builds surfaces and how it responds when those surfaces are activated. They are part of the material from which inflammation begins and part of the material from which it can later be resolved. They contribute flexibility, but they also introduce oxidation risk. They expand signaling possibility, but they also require structural balance and protective context. Once this is understood, the usual public conversation begins to look incomplete. The important question is no longer whether omega-3 is good or omega-6 is bad. The important question is what kind of body these fats are helping to build, what kinds of signals that body is prepared to generate, and whether its membranes have been remodeled toward proportionate response or toward chronic biochemical overreaction. Polyunsaturated fatty acids as signal substrates. Polyunsaturated fatty acids are not important only because they become part of membranes. They are important because once they are built into membranes, they create a stored substrate pool from which signals can later be generated. This gives them a role that is structurally deeper and temporally longer than most signaling molecules. A cytokine or neurotransmitter is often synthesized, released, degraded, and cleared within a relatively narrow window. A polyunsaturated fatty acid can sit inside a membrane phospholipid for extended periods, shaping membrane behavior quietly until a stimulus arrives that causes it to be released and converted into active mediators. That means the body stores part of its future signaling language in its membranes. Signal potential is built into tissue architecture before the signal is ever needed. The membrane is therefore not only an interface and not only a structural boundary. It is also a reservoir of conditional instructions waiting for the right trigger. This matters because signaling does not begin at the moment of conversion. It begins earlier at the level of substrate selection and tissue incorporation. When a membrane is enriched with certain polyunsaturated fatty acids, it is being preloaded with certain signaling possibilities. Arachidonic acid built into phospholipids creates one kind of inflammatory and vascular response potential. EPA and DHA built into phospholipids create another. These are not abstract biochemical options sitting in a textbook. They are physical realities built into tissue. When phospholipase activity is triggered by injury, infection, receptor activation, oxidative stress, or mechanical disruption, those stored fatty acids are released from the membrane and handed to enzymatic systems such as cyclooxygenases, lipoxygenases, and cytochrome-dependent pathways. What comes out next depends partly on what was sitting in the membrane in the first place. A cell cannot generate a given class of lipid mediator in meaningful amounts if the substrate pool for that class has not been built into its surfaces over time. This is one reason polyunsaturated fatty acids occupy such a powerful position in inflammatory biology. They do not just influence signaling from the outside by arriving in circulation. They influence signaling from the inside by being stored within the very structures from which signaling is launched. Arachidonic acid released from membranes can be converted into prostaglandins, thromboxanes, leukotrienes, and other eicosanoids that help coordinate acute inflammatory recruitment, vascular changes, pain signaling, platelet activity, and local defense. EPA can enter related pathways and shift some of those outputs while also giving rise to E-series resolvins. DHA can contribute to membrane structure and, once released, serve as substrate for D-series resolvins, protectins, and marazins, which participate in resolution, tissue protection, phagocytic efficiency, and restoration of order after inflammatory work has been done. The point is not that one substrate family is simply inflammatory and another is simply anti-inflammatory. The point is that polyunsaturated fatty acids supply the raw material from which inflammatory sequence is written. They influence how the response begins, how intensely it develops, and whether the system has the substrate available to move toward completion instead of remaining trapped in activation. Because of this, polyunsaturated fatty acids should be understood as conditional signaling material rather than passive dietary fats. Their double bonds give them flexibility within membranes, but those same double bonds also make them chemically reactive enough to participate in rapid enzymatic transformation when released. This is part of their biological value. The body uses their reactivity to generate fast, local, potent messages. But that same feature also creates vulnerability. A membrane heavily enriched in polyunsaturated material is carrying a larger reservoir of signal substrate, yet is also carrying a larger reservoir of oxidation-sensitive matter. Under well-regulated conditions, this allows dynamic signaling and adaptive response. Under poorly regulated conditions, especially when oxidative stress is high and structural protection is weak, those same polyunsaturated lipids can become sources of peroxidative injury, reactive aldehyde formation, distorted membrane behavior, and signaling spillover. The The same chemistry that makes them useful as signal substrates also makes them dangerous when control is lost. This is why substrate balance matters so much. The body is not just choosing whether to signal, is choosing from which stored materials signaling will emerge. If membranes are dominated by substrates that favor strong initiation chemistry, then the body will tend to enter inflammatory response with that material. If membranes are remodeled to include more substrates that support mediator classes involved in resolution and restoration, the body gains access to a different downstream language once activation begins. This does not mean structure disappears once signaling starts. Structure remains upstream the entire time, because the membrane is where the substrate was housed, and the membrane's composition determines the probabilities of future mediator production. A tissue that has been rebuilt under chronic inflammatory pressure may therefore become easier to activate not only because receptors are altered, but because its stored substrate reservoir has also shifted. The next signal is shaped by the last remodeling cycle. Polyunsaturated fatty acids as signal substrates also help explain why chronic disease often has a delayed, cumulative character. A person does not need an immediately obvious inflammatory crisis to be changing their future inflammatory behavior. Repeated dietary exposure, repeated oxidative injury, repeated phospholipid remodeling, and repeated immune activation can gradually alter the substrate landscape sitting in membranes across many tissues. Over time, that changes how the body is primed to respond. One tissue may become more likely to generate pain-amplifying mediators. Another may become more likely to sustain vascular irritation. Another may remain poor at generating the mediator profile required for efficient resolution. The person experiences the output as chronic inflammation, poor recovery, recurrent symptoms, or exaggerated sensitivity, but underneath those patterns is a membrane-level change in what signaling material is available when the next trigger arrives. The body carries its recent history inside its phospholipids. This also clarifies why the phrase inflammatory fat is often too blunt to be useful. A polyunsaturated fatty acid does not carry a moral identity. It carries structural and signaling potential. Arachidonic acid is necessary because acute defense requires initiation chemistry. EPA and DHA are important because control over inflammatory timing also requires access to substrate that can help shift the sequence toward resolution. The real issue is whether the stored substrate pool is proportionate to the demands being placed on the body and whether the broader structure can handle the reactivity built into those lipids. A membrane overloaded with highly oxidizable polyunsaturated material in a high-stress, poorly protected environment may become volatile. A membrane with too little dynamic signaling substrate may become structurally durable but less capable of proper adaptive signaling. Biology is not trying to maximize one category. It is trying to maintain a signal substrate pool that permits precise activation and clean exit without chronic overshoot. The importance of polyunsaturated fatty acids as signal substrates reaches beyond classical inflammation. These lipids also influence vascular tone, platelet aggregation, endothelial behavior, nociception, immune cell chemotaxis, barrier repair, microglial activation, and multiple forms of intracellular stress signaling. Even when they are not being directly released into well-known mediator pathways, their presence in the membrane changes the local environment in which receptors and enzymes operate. That means they participate in signaling both by becoming mediators and by shaping the surfaces through which mediator-generating events are interpreted. Their role is therefore double-layered. They are stored instruction sets, and they are part of the physical medium through which those instructions are triggered. Seen clearly, polyunsaturated fatty acids are not merely fats with multiple double bonds. They are reserved signaling substrate built into living structure. The body uses them to preload future inflammatory, vascular, neural, and reparative responses into its own membranes. That is why they matter so much in a book about omega-3, omega-6, and resolution biology. They explain how diet becomes tissue, how tissue becomes signal, and how signal becomes behavior. Long before a symptom appears, the substrate pool is already being written. Long before inflammation begins, the membrane has already decided what kind of language that inflammation will have available. Essential fatty acids and their pathways. The term essential fatty acid is often repeated without explaining what makes a fatty acid essential in the first place. A fatty acid is called essential when the body cannot synthesize it de novo and therefore must obtain it from diet. In humans, this applies to two parent polyunsaturated fatty acids, linoleic acid, the primary omega-6 essential fatty acid, and alpha-linolenic acid, the primary omega-3 essential fatty acid. The body can elongate and desaturate these parent molecules into longer chain derivatives, but it cannot create the omega-6 or omega-3 double bond pattern from scratch because it lacks the enzymatic machinery to insert double bonds beyond certain positions in the carbon chain. That limitation forces dependence on dietary intake. Essential does not mean that every downstream product will be produced in ideal amounts or that intake of the parent fatty acid automatically guarantees optimal tissue composition. It means the pathway has to begin with raw material the body cannot manufacture on its own. Once these parent fatty acids enter the body, they do not remain metabolically static. They enter elongation and desaturation pathways that can convert them into longer and more functionally specialized molecules. Linoleic acid can move through a sequence involving gamma-linolenic acid, dihomo-gamma-linolenic acid, and eventually arachidonic acid. Alpha-linolenic acid can move through stearidonic acid, icosapentaenoic acid, EPA, docosapentaenoic acid, and ultimately DHA through a more complex sequence that includes elongation, desaturation, and peroxisomal steps. These pathways matter because the parent essential fatty acids are not the end of the story. The longer chain derivatives are often the tissue-active forms most directly involved in membrane composition, inflammatory mediator production, neural structure, retinal function, and resolution biology. What the body can do with essential fatty acids, therefore, depends not only on intake, but on pathway efficiency, tissue demand, and metabolic competition. That competition is one of the most important features of these pathways. Omega-3 and omega-6 families share parts of the same enzymatic machinery, especially desaturase and elongase systems. This means the body is not processing each family on separate rails. They interact. High flux through one family can affect the handling of the other, not because one is inherently illegitimate, but because both are drawing from overlapping conversion capacity. In practical terms, the relative availability of linoleic acid and alpha-linolenic acid influences how much pathway attention each family receives. This is one reason simple dietary presence is not enough to explain tissue outcomes. A person may consume some alpha-linolenic acid, but if conversion pressure, competition, or metabolic conditions limit downstream production, tissue EPA and DHA status may remain modest. The pathway exists, but its throughput is not unlimited. The omega-6 pathway deserves precision because it is often discussed only through the lens of excess inflammation. Linoleic acid is the dietary starting point, but its biological significance depends on what happens after incorporation and conversion. Through desaturation and elongation, it can contribute to the formation of arachidonic acid, which then becomes a major membrane substrate for eicosanoid production. Arachidonic acid is central to acute inflammatory signaling, vascular control, pain pathways, and multiple forms of local immune activation. It is not a pathological accident. It is part of the body's normal capacity to respond rapidly to tissue disruption and threat. The problem begins when pathway dominance, membrane enrichment, and inflammatory conditions together bias the organism too heavily toward initiation chemistry without equally competent termination and repair. In that case, the omega-6 pathway is not wrong, but it becomes overrepresented in a system that has lost proportionality. The omega-3 pathway is often misunderstood in the opposite direction. Alpha-linolenic acid is the essential dietary precursor, but the longer-chain products EPA and DHA are the forms most directly associated with membrane remodeling, neural tissue composition, retinal structure, and specialized pro-resolving mediator generation. The conversion from alpha-linolenic acid to EPA is limited in many humans, and conversion to DHA is more constrained still. This is why the distinction between precursor omega-3 and tissue-active omega-3 matters. A diet may technically contain essential omega-3 while still supplying relatively little preformed EPA and DHA to tissues. From a pathway perspective, that means the organism has the raw starting material but may not be generating downstream products in the quantity or distribution needed to significantly reshape membrane composition or signaling capacity. Essentiality alone does not guarantee sufficiency at the endpoint. These pathways also operate within tissue-specific priorities. The body is not trying to convert every essential fatty acid into the same downstream product everywhere. Some tissues require more arachidonic acid for acute signaling readiness. Others require more DHA because membrane curvature, excitability, photoreceptor function, or neural stability depend on it. The liver plays a central role in handling, modifying, packaging, and redistributing fatty acids, but tissue uptake, remodeling, and retention determine where those molecules ultimately matter. A pathway on paper is therefore not the same as a pathway expressed in living tissue. The body does not simply manufacture EPA, DHA, or arachidonic acid and leave them floating in abstraction. It incorporates them into phospholipids, stores them in particular membrane pools, and then draws on those pools later under stress, signaling demand or structural turnover. This is one reason essential fatty acid pathways have to be understood as both structural and signaling systems at once. The pathway does not end when a fatty acid reaches its longer chain form. That product then enters membranes, changes membrane behavior, and becomes part of the substrate reservoir for future mediator synthesis. Arachidonic acid stored in phospholipids later becomes available for prostaglandin, thromboxane, and leukotriene production. EPA and DHA stored in phospholipids later become available for alternative eicosanoid profiles and for specialized pro-resolving mediator families. The pathway therefore determines not just what the tissue is made of today, but what kinds of inflammatory and reparative language it will be able to generate tomorrow. Essential fatty acid metabolism is stored future behavior. The oxidative vulnerability of these pathways also has to be considered. As the fatty acids become more unsaturated, they become more chemically flexible, but also more susceptible to peroxidation. This gives the body a trade-off. Longer-chain polyunsaturated products expand structural nuance and signaling capacity, but they also require better containment, better antioxidant protection, better membrane repair, and better control over inflammatory timing. A tissue enriched in highly unsaturated products without adequate protection is carrying more signaling potential and more chemical fragility at the same time. That is why pathway support cannot be reduced to the idea of making more long-chain polyunsaturated fatty acids at all costs. The question is whether the organism can incorporate and protect them within a stable architecture. Seen clearly, essential fatty acids and their pathways are not just nutrient conversion charts. They are the route by which diet becomes membrane composition, and membrane composition becomes inflammatory potential, neural behavior, vascular signaling, and resolution capacity. Linoleic acid and alpha-linolenic acid are essential because the body cannot begin these families without outside input, but what happens after intake depends on conversion, competition, tissue demand, oxidative context, and long-term remodeling. The real significance of these pathways is not that they turn one fatty acid into another. It is that they determine what kind of body gets built from the input. They determine which substrates are stored in membranes, which mediator families are available during stress, and whether the organism is being equipped more heavily for initiation, for adaptation, or for balance completion. Why the body requires both? The body requires both omega-3 and omega-6 because they are not interchangeable and they do not build the same signaling and structural possibilities. Each family contributes a different kind of material to membranes and a different set of downstream options once those membranes are activated. The mistake is to imagine that one family is the legitimate one and the other is a biological error. That is not how the system is built. The body needs initiation as well as resolution. It needs dynamic signaling as well as controlled shutdown. It needs membrane flexibility, tissue-specific responsiveness, vascular control, immune recruitment, neural function, growth, repair, and protection against overreaction. No single fatty acid family can carry all of those requirements alone. The body uses both because it is not trying to create one uniform lipid effect. It is trying to build a signaling-capable organism that can enter stress responses when necessary and leave them when appropriate. Omega-6 is required because acute biology must be able to mobilize quickly. Arachidonic acid-derived signaling is part of how the body generates pain sensitivity when tissue is damaged, changes vascular behavior when injury occurs, recruits immune cells when threat is detected, and coordinates local defense when disruption has to be contained. These are not optional functions. A body that could not initiate inflammation, alter blood flow, activate platelets, or recruit defensive cells would not be healthier. It would be less able to survive ordinary challenges. Omega-6 derived pathways are part of how the body responds with speed and force when a rapid local reaction is needed. That role becomes dangerous only when it becomes disproportionate, prolonged, or insufficiently opposed by pathways that help complete the response once its useful phase is over. Omega-3 is required because initiation alone is not enough. A body that can start inflammation but cannot guide it toward completion becomes trapped in its own defense chemistry. EPA and DHA matter because they alter membrane composition and because they provide substrate for mediator families involved in de-escalation, debris clearance, tissue protection, and restoration of order after inflammatory work has been done. This does not make omega-3 a passive calming agent. It makes it part of the machinery that prevents acute defense from becoming chronic damage. The body requires omega-3 because tissue protection, neural stability, membrane behavior, and inflammatory resolution all depend on having access to substrate that does more than intensify response. Without omega-3, the organism loses part of its ability to finish what omega-6 helps begin. The structural role also makes both families necessary. Membranes are not built from one kind of fatty acid because different tissues require different physical behavior. Some membranes need rapid signaling flexibility. Some need specialized curvature. Some need highly tuned excitability. Some need a signaling reservoir rich enough to support local inflammatory language. Others need a stronger contribution from lipids that support resolution-oriented mediator production or neural membrane behavior. DHA, for example, is especially important in tissues such as the brain and retina because those membranes require specific physical properties that shorter or less unsaturated fatty acids do not reproduce in the same way. Arachidonic acid remains important in tissues that need immediate signaling readiness. The body requires both families because it is solving multiple structural problems at once, not building one generic membrane repeated across all tissues. Both are also required because signaling in real physiology is sequential. Acute initiation without later resolution is destructive, but resolution without the ability to mount a proper early response is also dysfunctional. The body has to move through phases. First, it must detect disruption, then contain it, then recruit the appropriate machinery, then clear what needs clearing, then stop the escalation, then rebuild. Omega-6 derived substrate contributes strongly to the early and middle phases of this sequence. Omega-3 derived substrate contributes strongly to the later phases and to the membrane conditions that make proportionate signaling more likely in the first place. The body therefore requires both because biological control depends on sequence, and sequence depends on having access to different classes of substrate at different stages of the same event. This is why the real problem is not the existence of either family, but the loss of proportion between them. If omega-6 derived structure and substrate dominate too heavily, the organism becomes better prepared to escalate than to resolve. If omega-3 availability is too low, the body loses part of its capacity to complete inflammatory events cleanly and to maintain certain specialized membrane properties over time. On the other hand, simply adding omega-3 without regard for oxidative stability, membrane protection, and the broader lipid environment does not automatically restore balance. The goal is not to replace one family with the other. The goal is to restore coordination so that initiation chemistry remains available when needed, resolution chemistry remains available when needed, and membrane structure remains capable of supporting both without falling into chronic volatility. The body therefore requires both omega-3 and omega-6 for the same reason it requires acceleration and braking, activation and restraint, entry into defense and exit from defense. These are not opposing moral categories. They are complementary parts of a controlled biological sequence. Omega-6 helps the organism respond with force. Omega-3 helps the organism recover with intelligence. Both enter membranes. Both shape signaling. Both become part of tissue behavior over time. Health depends less on choosing sides than on rebuilding the relationship between them so the body can defend itself without becoming trapped in defense. The myth of good versus bad. One of the most damaging habits in nutrition is the tendency to sort molecules into moral categories. A fat becomes good, another becomes bad, and the discussion ends before physiology has even begun. That habit makes marketing easier, but it makes understanding harder. The body does not use lipids according to moral identity. It uses them according to structure, context, timing, tissue demand, enzymatic access, oxidative environment, and the sequence of events the organism is trying to carry out. A fatty acid can be useful in one role and harmful in another. It can support membrane function in one tissue and become excessive signaling substrate in another. It can be necessary in acute defense and destructive in chronic dysregulation. Once that is understood, the good versus bad frame starts to collapse because it was never precise enough to describe lipid biology in the first place. Omega-6 is one of the clearest examples of this distortion. It is often treated as the villain because arachidonic acid-derived mediators participate in inflammation, pain, platelet activity, and acute vascular response. But those pathways exist for reasons. The body has to be able to respond when tissue is damaged, when pathogens are present, when bleeding has to be controlled, or when local defense must be mobilized quickly. Without omega-6 derived signaling, the organism would not become cleaner or calmer. It would become less competent. The real problem is not that omega-6 exists or that it participates in inflammatory signaling. The problem is that in many modern conditions, omega-6 derived structure and signaling become overrepresented in a body already under chronic oxidative, metabolic, and inflammatory strain. The issue is not moral corruption of one fat family. The issue is disproportion inside a system that has lost control of sequence. Omega-3 is distorted just as badly, but in the opposite direction. It is turned into the heroic fat, the universally beneficial input that can be added to almost any health problem, as though it carries its own built-in corrective intelligence. That is too simple. Omega-3 can be extremely important, but its value depends on what kind of membrane it enters, what oxidative conditions surround it, how much of it is actually incorporated into tissue, how much competing substrate is already present, and whether the body has the enzymatic and structural capacity to use it well. EPA and DHA can support membrane behavior and contribute to resolution-oriented signaling, but they are still highly unsaturated molecules. In unstable oxidative environments, that unsaturation can also become a liability. That does not make omega-3 bad. It means even a valuable lipid can create vulnerability if it is introduced into the wrong architecture without the stability required to protect it. This is why isolated praise and isolated blame both fail. Arachidonic acid is not bad because it helps generate inflammatory mediators. Those mediators are part of normal defense. DHA is not good simply because it's found in neural tissue and resolution pathways. It still has to be handled, incorporated, protected, and balanced. Saturated fats are not bad because they reduce membrane fluidity relative to polyunsaturated fats. In many contexts, they contribute structural stability and resistance to peroxidative damage. But that does not mean all membrane behavior improves by maximizing rigidity. Cholesterol is not bad because it appears in cardiovascular discourse as a risk marker. It is also a membrane organizer and steroid precursor, without which cellular architecture and endocrine biology would collapse. The body does not experience these molecules as brand identities or online arguments. It experiences them as materials with specific physical consequences. The good versus bad framework also fails because it ignores role switching. The same lipid can serve different purposes depending on state. A polyunsaturated fatty acid stored in a membrane today may later become inflammatory or pro-resolving substrate tomorrow. A triglyceride stored as fuel can later influence signaling through the conditions of its mobilization. A structural lipid can become part of a signaling cascade once released. A signaling event can alter future structure by changing how membranes are remodeled after stress. In biology, lipids are not trapped in one category. They move between architecture, fuel handling, and communication. Once molecules are understood as role-shifting components inside a dynamic system, simplistic labeling becomes less useful. The real question is never whether a lipid belongs on the good list or the bad list. The real question is what it is doing in that tissue, at that time, under those conditions. This moral framing also hides one of the central truths of chronic disease, which is that harm often emerges from imbalance, misplacement, overrepresentation, or failed sequencing rather than from the mere presence of a molecule. The body does not usually break because it encountered one evil nutrient in isolation. It breaks because structural ratios shift, signaling timing deteriorates, membranes become easier to oxidize, mitochondrial handling becomes less efficient, and inflammatory pathways lose their ability to end cleanly. In that situation, even normal biological components can participate in pathological outcomes because they are being used inside a disordered system. Arachidonic acid can contribute to chronic inflammation when resolution fails. Highly unsaturated omega-3 incorporation can become fragile when antioxidant protection is weak. Saturated structures can become limiting if adaptability is lost in certain membrane contexts. None of this is captured by calling one molecule clean and another toxic. The system has to be read as a system. The language of good and bad also encourages people to think in terms of elimination instead of architecture. They look for the enemy fat to remove or the hero fat to add, as though the body were waiting for a single dietary verdict. But the organism is trying to maintain surfaces, gradients, receptor function, barrier control, signaling reserve, mitochondrial performance, and immune timing all at once. That means the relevant question is compositional. What kind of membrane is being built? What kind of signaling reservoir is being stored? What kind of oxidative burden is the system carrying? What kind of structural protection surrounds highly reactive lipids? What kind of inflammatory sequence is the body equipped to generate and complete? Those questions are harder than simply asking which fat is good, but they are the questions that actually correspond to physiology. This is especially important when discussing omega-3 and omega-6 together. The body does not require a hero and a villain. It requires a relationship. Omega-6 helps create rapid local response capacity. Omega-3 helps shape membrane behavior and expand access to mediator families involved in completion and repair. Both are necessary. Both can become problematic in the wrong context. Both derive their meaning from proportion, tissue placement, and system state. The problem begins when one family dominates structural incorporation and downstream signaling to the point that the organism becomes better at escalation than at closure, or when the attempt to correct that imbalance ignores the oxidative and architectural conditions required to make the correction hold. The issue is never simply one good input rescuing the body from one bad input. It is whether the lipid environment has been rebuilt into something coherent enough to support proportionate biological behavior. A more honest framework is to stop asking whether a lipid is good or bad and start asking what problem it solves, what liability it introduces, and what conditions determine which side of that balance becomes dominant. Every meaningful lipid does both. Polyunsaturated fats provide signaling flexibility and mediator diversity, but they also introduce oxidation risk. More saturated structures provide durability and resistance to peroxidation, but they can reduce flexibility if not balanced appropriately. Cholesterol stabilizes and organizes membranes, but excessive mismanagement of cholesterol traffic within broader metabolic dysfunction can still reflect deeper systemic problems. Biology does not eliminate trade-offs. It manages them. A useful nutritional framework has to do the same. The myth of good versus bad survives because it is emotionally satisfying. It offers a simple map in a system that is actually conditional, layered, and physically constrained. But the body is not asking for moral clarity. It is asking for proportion, sequence, and structural coherence. Lipids are not saints or villains. They are materials. Some build faster signals. Some build stronger surfaces. Some provide substrate for escalation. Some provide substrate for resolution. Some do more than one of these at the same time. Health depends on how they are arranged, not how they are judged. Once that becomes the frame, the conversation improves immediately. It stops being about choosing the righteous fat and starts being about rebuilding the body's lipid architecture so that defense, energy, and recovery can occur without turning against the organism itself.